I'm Dr. Kokon Kumar Dotto. I have a Christian name, Dr. David Boone. I'm currently working as assistant professor in the Department of Biochemistry and Molecular Biology in Bangabandhu Sheikh Mujibur Rahman Science and Technology University in Gopalganj, Bangladesh. The title of my discussion is TXNIP or TBP-2 or VDUP1 plays key role in sting mediated immune responses. Sting stands for stimulator of interferon genes. Sting is encoded by the TMEM173 gene and also called TMEM173. TMEM stands for transmembrane protein. The other names for sting are MPYS, Meta, and Aries. Sting induces the production of type 1 interferon, interferon alpha, and interferon beta, especially when microbes like viruses, bacteria, parasites, and others invade the human body and infect the cells. Interferons are also strong cellular weapons against the tumor and cancer cells inside the body. Formation of unfolded protein aggregates and unfolded protein responses are associated with microbial infections like viral infections in most of the cases. Endoplasmic reticulum, ER, is the intracellular compartment where proteins are usually gate folded in proper functional conformations. Protein producing factories, ribosomes, are found attached with the endoplasmic reticulums. Endoplasmic reticulums play important roles in calcium homeostasis, and endoplasmic reticulum are very responsive organelle under redox stresses and even can induce the upregulated production of reactive oxygen species ROS to create an stressful environment which is able to inflame the mitochondria to initiate inflammatory responses. Molecular chaperones play important roles in both the generation and to mediate the subsequent effects of the unfolded and misfolded protein aggregations. Golgi apparatuses are the sites where translated proteins are modified and packaged to deliver in different locations and in the functional destinations, both inside the cells and outside the cells. Deregulations in endoplasmic reticulum functions result in unfolded and misfolded protein productions. Golgi apparatuses function in close proximity of the endoplasmic reticulum. After modifications inside the endoplasmic reticulums, proteins usually enter the Golgi apparatuses in the exit points. Various stimulants and causatives are seen to induce the production of unfolded and misfolded proteins. Viruses usually induce the enhanced production of unfolded and misfolded proteins from the endoplasmic reticulums for their survival, replications, or multiplications and clearances. Viruses are seen to multiply in specific locations and environments inside the living cells which are essentially rich with aggregations and autophagy-related proteins. Unfolded and misfolded proteins, on the other side, induce mainly mitochondria-regulated inflammatory responses that usually destroy the viruses. The other wing of the host responses against the viruses is mediated by interferons, which also links with the production of different cytokines and with the inflammatory process ultimately. Unfolded and misfolded proteins load actually activate different pathways in the host. Some of them works to benefit the viruses, and the others eventually try to eliminate them from the body. Viruses and other pathogens usually try to hijack and to modulate the components and the mechanisms that act as the potential danger for themselves. To mention here that the mitochondria are now increasingly evident as a central regulator of the body's protection mechanisms against both the external and the internal threats, besides their well-known roles in the power or ATP generation for the cells and the body as a whole. Unfolded protein responses or endoplasmic reticulum stress connects with the mitochondrial events to activate the protective inflammatory processes and other protective mechanisms, including the interferon IFN responses, by mainly upregulating the level of thyroidoxine interacting protein TXNIP. Inositol recording enzyme 1 IRE1 is a sensor of endoplasmic reticulum stress. A microRNA 17, usually suppresses the expression of thyroidoxine interacting protein TXNIP. When IRE1 is activated, it inhibits microRNA 17, possibly via read, that results in upregulated expression of TXNIP. Protein kinase R, PKR, like endoplasmic reticulum kinase, PARP, also works as a sensor for the ear stress. PARP increases TXNIP expression via the induction of transcription factors, carbohydrate responsive element binding protein, CREBP, and activating transcription factor 5 or ATF5. The events inside the endoplasmic reticulum are strongly associated with the redox status. Free radicals are produced in the endoplasmic reticulum during the protein folding process. High load of proteins in the endoplasmic reticulum also increase the free radical production and oxidative stress that induce elevated expression of thyroidoxine interacting protein TXNIP. Thyroidoxine interacting protein TXNIP then translocates in the mitochondria. In the mitochondria, TXNIP blocks the free radical scavenging activities of thyroidoxine 2, TRX2, and leads the increased accumulation of ROS and oxidative stress. High level of presence of TXNIP in the mitochondria cause different degrees of damages to the mitochondria. Different pathways are activated depending on the extent of damages to the mitochondria. One of those pathways activate the inflammation and the inflammatory process. TXNIP associates with and activate NLRP3 inflammation in the mitochondria. TXNIP also interact with and activate NLRP3 inflammation in the cytoplasm. Activated NLRP3 inflammasome complex induce the production of interleukin 1 beta by activating caspase enzymes. Interleukin 1 beta subsequently initiates a cascade of inflammatory events, including the production of different cytokines. TXNIP mediated mitochondrial damages also increase the levels of free radicals in the cytoplasm and introduce the presence of mitochondrial DNA in the cytoplasm or cytosol. Stimulator of interferon genes, STING, becomes activated when it recognizes the presence of mitochondrial DNA in the cytosol. Mitochondrial damage and DNA release also activate another molecule, cyclic GMP AMP synthase, C gas, which in turn catalyzes the generation of cyclic guanosine monophosphate, adenosine monophosphate, cyclic GAMP ligand that stimulates the STING. STING can also be activated by interacting with the RNAs of some viruses. When STING is activated, it translocates and induces the production of interferons. Interferons are the first line defense of the body against viral infections. Interferons secreted from the infected cells alert the neighbors to heighten their defenses against the viruses. Interferons also stimulates immune cells like natural killer cells or NK cells, macrophages, and others. Interferons also induce the expression of hundreds of genes which help to fight the viruses. Viruses usually, in the first hand, try to modulate the production of interferons for their benefit. It would be interesting here to mention that the deregulation in the expression of both thyroidexine interacting protein TXNIP and interferons IFNs have been reported in novel coronavirus 2019 or COVID-19 infections in humans and human cell lines. To admit here in the conclusion that there are other mechanisms known to the scientists that can also activate STING and interferon production in the viral infections and in other diseases. The hints and mechanisms are discussed here in the light of the very recent advances in these frontiers of immunology, which may seem different from the previously known pathways and mechanisms. Thank you, everybody. Good day.